Okay, we are now moving on to carbon reaction. This picture shows the process of photosynthesis. During the light reaction, you get an ADPH and ATP from the moving of an electron that gets excited by the photon from the light. And these two products, ATP and NADPH, will be used to fix carbon dioxide and to become sugar in the carbon reaction, or in this case, it's called Calvin-Benson cycle. So the carbon reaction was long thought to be an independent of light, and it's called dark reactions. However, the substrate of the carbon reaction was actually depend on the light, and moreover, many enzymes in the carbon reaction is activated by light. So we better call it carbon reaction than dark reaction. So let's move on to here. I like to change my pointer to the laser. So in carbon reaction is basically Calvin-Benson cycle. We have three phases. Actually, in your previous knowledge, you may know it as just Calvin cycle, but later it's better call it Calvin-Benson or Calvin-Benson-Becham cycle, because later people discover that it's not only Marvin Calvin, that discovered this cycle, but it's also his colleagues make a lot of contribution, and he's Benson. So later we put the two names of two persons together, the Calvin-Benson cycle. So let's go back to our lecture. So Calvin-Benson cycle has three phases. The first one is carboxylation, as the name suggested. Carboxylation is the carbon fixing. We start by using the carbon 5 molecules, ribulose 1,5 bisphosphate, or RUBP in short. And these 5 carbon molecules will be used to fix carbon dioxide, which has only 1 carbon. And 5 plus 1 equal to 6, right? But actually, the 6 carbon molecule is not stable. So the 6 carbon molecule is splitting into 2. You will get 2 molecules of 3 carbon compound. It's called 3-phosphoglycerate. The next phase is to make the 3-phosphoglycerate more energy. By having more energy, it means having more electron. More electron is mean more reduced. And the next phase is called reduction. And this reduction means reduce in oxidation number, getting more electron. How to do that? By using ATP and NADPH, the products of the light reaction. And by that, you will get the same amount of carbon in the molecules, but this carbon has higher energy. This molecules is called glyceraldehyde 3-phosphate. It's a triose, which means a sugar with 3 carbon. Attached to it is phosphate that came from ATP. And from here, if you make a lot of glyceraldehyde 3-phosphates, some of that will need to be put back together to form the ribulose 1,5 bisphosphate again in order to regenerate, in order to recycle the carbon backbone. So in this phase is called regeneration. Again, it used ATP. So, and some of the glyceraldehyde 3-phosphate right here can be put together into bigger molecules of sugar. It's right now it has only 3 carbon, right? It can be combined with another 3 carbon to become 6 carbon sugar. For example, to become glucose, to become sucrose, and then those glucose can be used as a substrate to make amylose, amylopectin in starch. It can be used to make sucrose, and this sucrose can be transported in the phloem. So sugar that can transport in the phloem is in the form of sucrose, is in the phloem transport. And this one will be stored as the polysaccharide in different tissue that cannot do photosynthesis. All right, let's go to more detail on carboxylation of ribulose 1,5-bisphosphate. This reaction is catalyzed by an enzyme called rubisco. Rubisco is the most abundant protein on Earth. So in not a muscle that the most abundant, but plants has a lot of this enzyme rubisco. Why? Because plants do a lot of photosynthesis, of course. And another reason is rubisco is not quite efficient. So you need a lot of rubisco to make sugar. If rubisco is efficient, you don't need that much. So you need just a little bit and then you get a lot of the products, right? Because rubisco is not only fixing carbon, in some situation, it also fixes oxygen. And by fixing oxygen, it stores the enzyme, make the enzyme stop working. Not only making the enzyme stop working, it also create a toxic products that the plant need to get rid of that products too. So we will talk about that more on the next video, I guess. So as I mentioned about the 3 carbon compound, 3-phosphoglycerate right here, this one is the first products of carbon fixation, and most of the plants will get the 3 carbon products. We call those plants C3 plants, but some plants 3 carbon compound is not the first products, but it's actually 4 carbon compounds. All right, so let's go to the reduction phase. The reduction phase is composed of 2 steps. The first one is consuming ATP. The second one, consume NADPH. ATP will provide energy and also the phosphate. Notice that this 3-phosphoglycerate, the beginning, has only 1 phosphate in the molecules. But after the first step by enzyme phosphoglycerate kinase, the enzyme name kinase is mean adding phosphate. Every time you see the word kinase, it means you adding more phosphate to the reactant or the substrate. So the products of 3-phosphoglycerate kinase is 1,3-bisphosphoglycerate. You have 2 phosphate. That means bis is mean 2, and the number on the beginning is the position of phosphate in the molecules. So the next step is to reduce by having an NADPH, and an NADPH can donate 2 electrons and it will become an ADP plus, and these 2 electrons will kick out 1 molecule of phosphate out, and you will get more reduced products, 3-phosphoglycerate. From here, you can see that you use more ATP and you use more NADPH, these two guys came from the light reaction. I forget to mention. This reaction is catalyzed by an enzyme glyceraldehyde 3-phosphate dehydrogenase. You see, this is the dehydrogenase is an enzyme involving in redox reaction, gaining or losing electron, as in the lab 7 metabolism is about the dehydrogenase that you have raw potato and cooked potato, and you put that together with methylene blue, and later you find out that the raw potato can change the color of methylene blue from blue to clearless because methylene blue receive electron from the hydrogenase, and methylene blue become reduced and it change the color. All right, so the last step is the regeneration of RUBP or ribulose bisphosphate, 1,5-bisphosphate, sorry. So the 5 of the 6 molecules of glyceraldehyde 3-phosphate can be combined into many steps and becomes the new ribulose 1,5-bisphosphate again. Where is it? I can't see. Anyway, this step involves 10 enzymes, and it is important for the continuous cycle of Calvin-Benson cycle. All right, this is the overall or overview of Calvin-Benson cycle. You start from RUBP, and then you get carbon dioxide, 3-phosphoglycerate, make it more powerful, and get glyceraldehyde 3-phosphate. If you start from 3 molecules of carbon dioxide and 3 molecules of RUBP, you end up with 6 3-phosphoglycerate and 6 glyceraldehyde 3-phosphate. One of the 6 glyceraldehyde 3-phosphate will be used to make glucose, and 5 of them will be used for regeneration of RUBP. During the regeneration, you spend more ATP. If you may recall, the electron transport chain in the light reaction. During the light reaction, you have the linear electron flow where you can make ATP and NADPH. And also, you also have the cyclic electron flow. You can see that cyclic electron flow give only ATP, and you combine that knowledge to this picture. During the Calvin-Benson cycle, you need 6 molecules of NADPH and 9 molecules of ATP. This means you need more ATP compared to NADPH. So it is making sense right now that the cyclic electron flow is quite important because it needs to make up the ratio of NADPH and ATP. If you have too much of ATP of NADPH and it cannot be used for Calvin-Benson cycle, NADPH has a very high reducing power. It can give electron to many components in the cells. Once those components get more electron, it is not a good thing because when they get electrons, they become more excited and it becomes toxic to the cells. So the plants need to balance those reducing power of NADPH by making more ATP. And then that can be used for fixing more carbon and fixing more carbon to become starch that quite safe and non-toxic and can be used for energy in the cells, right? So I will stop right here, and in next video I will talk about Rubisco and how it's become the most abundant enzymes on Earth.